In this video, we're going to talk about the steps of the sliding filament theory. As we've been talking about, a key thing to remember is that proteins change shape when things bind to them. The myofilaments, actin and myosin, are proteins, so that means that when things come into the picture that bind to them, there's going to be changes in their shape, resulting in interactions which create the sliding filament theory. Because skeletal muscles are voluntary, there needs to be an interaction between the nervous system and the skeletal muscle cells. That is called the neuromuscular junction. So between a motor neuron, for right now it's just important that you recognize that this is a link to the nervous system, and the myofiber, the skeletal muscle cell, that junction is called the neuromuscular junction. From the motor neuron is going to be a neurotransmitter called acetylcholine. It's easier to abbreviate this as capital A, capital C, lowercase h, and refer to it possibly as ACH or acetylcholine. Acetylcholine is a neurotransmitter, which means that it's a messenger between the nervous system and the sarcolemma, which is the skeletal muscle cell's membrane. When the acetylcholine binds to the sarcolemma, so when it touches that cell membrane, it's going to trigger an action potential, which is represented by those arrows hopping down the cell membrane. Because the sarcolemma goes strictly around the muscle cell, we need something to bring it into the cell. That's where all of those myofilaments are that are going to be interacting with each other. So the muscle cell uses a structure called T-tubules to bring the action potential down and into the cell. So it's coming down through little openings on the sarcolemma, and those T-tubules go down and throughout the cell. Another important thing about the location of the T-tubules is its overlap with the SR, the sarcoplasmic reticulum. The sarcoplasmic reticulum is a structure that surrounds every myofibril. It's what we've kind of called the puffy looking thing. It's puffy in a sense because it's storing calcium ions. Once that action potential comes zipping through the T-tubule and it hits the point where the sarcoplasmic reticulum is overlapped, that gives it an impulse to release calcium ions from the sarcoplasmic reticulum. Keep in mind, proteins are going to change shape when things bind to them. Those calcium ions are going to bind to troponin. Troponin is one structure on the actin myofilament. The picture on the left here shows your actin strand. So actin is represented by kind of a coiled band of, looks like beads. Troponin is this gold-ish structure. It's kind of the pin that holds tropomyosin, this purple rope-like structure, in place over the active sites. When calcium is going to attach to the troponin, in this right-hand picture, we can see that the active sites, represented by the little blue eyes here, those are going to be exposed because troponin changed its shape when calcium bound to it, and that change in shape pulled that myosin rope away, and now I have my active sites exposed. Active sites are the spots where myosin wants to get to, and once they are exposed, the myosin is going to form what is referred to as a cross bridge. Cross bridge simply means that there's a connection between the actin strand and the myosin head. Myosin is able to form this cross bridge because of a form of stored energy. At the end of the last contraction cycle, myosin is left with its head pointing away from the middle of the sarcomere with ADP, adenosine diphosphate, and an extra phosphate group. Adenosine diphosphate plus phosphate is a form of split energy. The energy has been produced because we broke the phosphate group off. That energy is stored in the myosin head and is going to be used to make that connection up to the cross bridge where phosphate will actually be released out into the sarcoplasm. And from here, the head is going to pivot towards the midline. That pivoting towards the midline or M line is called the power stroke. This simply means that it is pushing the actin filament towards the middle of the sarcomere. Because the actin myofilaments are being pushed towards the middle of the sarcomere, that means that the result is going to be a shortened sarcomere. Your sarcomere is brought closer together, which would be something you would witness in your muscles getting shorter as it contracts. At the end of the power stroke, the adenosine diphosphate, the ADP group, is going to be released out into the sarcoplasm as well. When something unbinds to that myofilament, it's going to change its shape. That shape change lends itself to the binding of something new, which will be a new ATP molecule. And that new ATP molecule makes the head release from actin's active site. This would be referred to as breaking the cross bridge. And the head would return back towards the outside. As it goes back to this state, it would return to the step at the beginning where ADP plus P were on the head with the stored potential energy, where we could again form another cross bridge, do another power stroke, have a new molecule of ATP bond to it, and go back and forth again and again and again. So that would keep going until there would be a lack of calcium ions or a lack of ATP, because then myosin couldn't detach itself from the actin myofilament. Or it will stop because there's no impulse. So a way to end skeletal muscle contraction is to get rid of the acetylcholine that's on the sarcolemma, because remember, that's what kind of triggered all of this in the first place. So getting acetylcholine off of the sarcolemma is going to end the action potential, meaning that there's no action potential going down that T-tubule and over the sarcoplasmic reticulum. If no action potential is hitting the sarcoplasmic reticulum, it's going to recapture all of the calcium ions, and including the ones that had been on actin and even those that had been on troponin. Since calcium is off of troponin, the active sites are covered up, meaning that myosin cannot bind with actin at all. So we're stuck in that stage where it has stored energy in the head, the adenosine diphosphate plus phosphate group. So whenever the active sites are exposed again, we can get going with cross bridge, power stroke, new ATP. So at this step, we would say that the muscle is relaxed, the sarcomere is lengthened, because there's no way for the actin filament to be pushed towards the middle if the myosin heads are not contacting it. That is the sliding filament theory. Just keep working with it. I know it's a lot, but get familiar with those structures, get familiar with that sequence.